Welcome to my poster, Computational Modeling of the Human Microbiome of the Respiratory Tract. My name is Andreas Dräger. I'm from the University of Tübingen at the Institute of Bioinformatics and Medical Informatics. As the interface between the human body and the outside world, the human nose and the respiratory tract harbor numerous bacterial species and are a vital target of virus particles. Many bacteria are commensals, but some are particularly life-threatening, such as Staphylococcus aureus, which no longer reliably responds to antibiotic treatment. The COVID-19 pandemic has also demonstrated the threat of virus infections. The mutual interactions between bacteria, their host, and viruses influence the risk of an infection outbreak. Consequently, altering the microbiome could pave the way toward novel therapeutic applications. However, a deep understanding of our inhabitants and their mutual interaction remains indispensable for its exploitation. So we have here assembled a unique priority list of the respiratory tract's 148 most relevant bacteria and systematically reconstructed genome-scale metabolic computer models of each of these species. To this end, we refined existing protocols and standard operating procedures for efficient model reconstruction while ensuring highest quality measures. We contributed to the definition of standard file formats for a graphical representation, such as SPGN and SPGNML, model distribution, such as SBML, and the benchmarking tool MeMode for quality assurance and quality control. We implemented software for efficient model simulation, such as SBSCL, the Systems Biology Simulation Core Library, including a web front end named SBML Web App. We have already reconstructed many bacteria, including Corynebacterium simulans, Dolichogranulum olympicum, Phenegoldia magna, Moraxella catahalis, Pseudomonas aeruginosa, Rothia mucoliganosa, Staphylococcus epidermidis, and 114 strain-specific models of Staphylococcus aureus by applying our modeling pipeline, followed by careful manual curation. Metabolic reconstructions of several additional microbes are in development. In addition, we refined modeling techniques for simulation of virus reproduction and applied them to COVID-19 to identify weak spots of the virus. The algorithm PyMCadre allows us to reconstruct human cell models for specific tissue types. We have also developed new methods to combine two or more models to simulate mutual influences while reconstructing further inhabitants in the tool NCMW. In conclusion, computational models provide a valuable basis for integrated wet lab, dry lab research projects. Their predictions can guide not only experimental work, but also identify new potential targets for the investigation of complicated mutual interactions between microbes, viruses, and their host. By validating model-derived hypotheses, it may become possible to drastically reduce the growth and colonization ability to threatening pathogens such as Staphylococcus aureus from the nose to the lungs. This project has laid the foundation for systematic model building, quality control, open distribution, validation, and simulation. The outcomes of this project support the search for novel antimicrobial therapies against multi-resistant pathogens or severe viral infections. Thank you for your attention.